So we'll start this session on improving targeted therapies with David Scadden. David's a professor of medicine at Mass General Hospital, and he'll be talking about what can stem cells teach us about cancer biology.
Thanks very much, Don. It's a great pleasure to be here, in particular to follow two of my mentors, and I have to say it's always a great pleasure and opportunity to have a chance to thank them for opportunities that were given to me. And I will give one Bruce Chabner anecdote, which is that when I first met Bruce, I had just come to the MGH, and Bruce was instrumental in my going to the MGH. And at the end of the meeting, he turned to me and he said, "So how can I help?" I thought, my goodness, that's extraordinary. I'd never heard that before, actually. And it really is that he characterizes the mentorship that he provides and the care he has for junior people, and I much appreciate that. So I'm going to tell you a little bit about some of the things that we've been working on, because I trained as a hematologist oncologist, but I focused mostly on stem cells and thinking about blood stem cells in particular, trying to understand the ways in which they are regulated, and then trying to use that information about normal cells to see if there are ways in which that can provide some new information about cancer cells, and to try to identify ways in which we can ultimately use that to develop novel strategies to treat hematologic malignancies. So first of all, my disclosures, which will indicate that you should discount everything I say. But let me just talk about one project in particular, which is the work really of one of postdocs in my lab, Stephen Sykes, who's done a tremendous amount of work on this story that I'll go through with you. And I have to say, Stephen has done such a great job of compiling this that I pirated most of these slides from him. Now, of course, no one works in isolation, and he had a great deal of help from other members, other trainees in my laboratory and in other laboratories, and in particular with help from the laboratories of Scott Armstrong, Gary while he was still here at Harvard, Rhonda Pino, a collaboration with Lars Bullinger, who is a geneticist working at the University of Ulm, and then with our colleague in clinical medicine, Eyal Attar. So the idea of thinking about using stem cells as a way to consider issues in cancer. I mean, one thing, the cancer stem cell hypothesis has taken a fair amount of hits and is certainly got lots of flaws, but I think it actually still provides us with at least some conceptual advantages if we use it to apply to cancer. And one is that the heterogeneity of cancer, we can think of if we think of cancer as a tissue, the way normal tissues are organized, that perhaps this heterogeneity does represent something of a hierarchy. And that within that hierarchy, there are subsets of cells that have particular features that render them particularly dangerous. That is, that there's a population of cells which has stem-like features. And stem cells have two cardinal features. One is the capacity to self-renew, which is not uniformly the case in cancer cells, but in a subset of cancer cells, that would be, of course, the cell population that would be most dangerous, if you will. But that these cells also have some capacity to undergo limited degrees of differentiation, and that this would be consistent with what a stem cell population would do. In addition, the stem cell notion is that stem cells are not totally cell-autonomous units, but rather they are regulated by a specialized microenvironment, a so-called stem cell niche. And this idea that if we understand cancer in terms of the subpopulation of cells that are the self-renewing population, that population, if it is following other stem cell rules, may have interactions with the microenvironment that can be useful. But I think this whole notion is one that really has the advantage that it provides for those interested in cancer is mostly that it gives us an opportunity to be able to look at normal cell populations that may be correspondent to some of the malignant cell populations and determine whether or not we can learn anything useful from the normal cells that will give us an advantage in evaluating the cancer cells. So I'll just mention one area that we've been particularly interested in is this issue of the niche. The tumor microenvironment has long been an issue of consideration in cancer biology, but we think in the hematopoietic system, by teasing apart different elements of the stem cell niche, we've been able to define that certain subpopulations of cells that represent early osteolineage cell populations that we found regulate the hematopoietic system, that maybe this is informative about how these cells may participate in contributing to a tumor microenvironment. And so work done by Mark Ramacher and Sid Mukherjee, with many others in the laboratory, indicated that they could modify a subpopulation of mature cells, which we had previously identified did not really contribute much to hematopoietic regulation. And if you perturb this population, you end up not having much of a hematopoietic phenotype. But if you take a more immature osteolineage population that we thought was more of a normal stem cell niche provider, that if you disrupt its function, you end up with a myelodysplastic phenotype and the emergence of an acute myeloid leukemia with new complex molecular abnormalities arising in that leukemia, suggesting that by understanding subpopulations of cells that participate in normal hematopoiesis, that maybe we can identify cell populations that are altered in the context of a dysplastic phenotype, or maybe perhaps supportive of the emergence of malignant cell populations in the context of leukemia. Now, applying this to the cells of the hematopoietic lineage itself, if we think about how hematopoiesis is organized, the concept that it is hierarchical with a stem cell population at the top of the cascade has been well defined experimentally. And we think by studying this population, we've been able to identify a number of pathways that are important in regulating its function. And the question is now, can we apply this to a population of leukemic cells, where we know that AML does not really typically descend from, arise in a stem cell, but that it arises in a more mature progenitor population, but that this progenitor population acquires stem cell-like features, that it does have the capacity to self-renew, it has a limited degree of differentiation. So we wanted to evaluate this in the context particularly of trying to solve an important problem in leukemia. And as we know, in the setting of CML, there's a very clear relationship between a particular genotypic abnormality and the disease. That's very different in the setting of AML, where there's tremendous heterogeneity in the cell populations, very complex genotypic abnormalities. And the idea of being able to define either a single molecular target or multiple molecular targets for the wide range of different genetic abnormalities seen in this tumor actually represents a complexity that's almost going to be impossible to solve. And so we asked the question whether or not there might be ways in which we can broadly dissociate the AML genotypic abnormalities and impose, if you will, a slightly simpler way of evaluating different subpopulations of leukemic cells. And so what we've done is then try to ask the question of not whether you can categorize AML based on its genetic alterations, but rather based on some of the signaling, some of the characteristic dependence on signaling mechanisms that we had defined in normal stem cells. And in particular, we wanted to look at the issue of AKT pathway, and we've now demonstrated that there are indeed subpopulations of cells that have high or low AKT activation. And I'll focus on our story, which now looks at the subpopulation with lower AKT activation. So the way to approach this is, of course, to think about the way in which the signaling occurs. And AKT signaling, as you know, is something that is a mediator of activation of, it gets activated by a number of different growth mediating signals. It has an impact on a number of downstream targets, very complicated, but we focused really on one particular area of emphasis, which is that of the interaction with FOXO, where activation of AKT phosphorylates FOXO and renders it inactive. That is, it prevents it from being able to enter the nucleus. So this is the pathway that we were particularly interested in particular because it had been demonstrated that FOXO is a key regulator of normal hematopoietic stem cell function. It is also, as we know, an important mediator of stress resistance. And stem cells generally have relatively high levels of FOXO, in part because it's hypothesized that maybe it provides them a particular ability to resist tissue stress. So we asked then, what's the status of FOXO in AML? And looking at a signature that had been generated by Zuzana Titova when she was in Gary Gilliland's lab at the hematopoietic stem cell signature of cells with or without FOXO, she identified that there is a characteristic set of genes that are on and off, and of course this is too complex to see in any detail, but that providing this as the algorithm for determining whether or not there's active or inactive FOXO pathway, and then applying this to AML, working with Lars Bullinger at University of Ulm, discovered that there are indeed ways in which you can subfractionate AMLs into those with low FOXO activity and those with high FOXO activity, again, according to the signature defined in a mouse model of FOXO in normal stem cells. So with that, it appeared that there were ways in which you could break down AML broadly into these two categories. And interestingly, it was a way in which you could break it down that seemed to be somewhat independent of the different genotypic abnormalities in leukemia. Clearly, there were some genotypes where there was very consistent low FOXO activity, but for the most part, it was across the different genotypic abnormalities that were seen. So it's exactly what we were looking for. Now, it did have some inverse correlations, and in particular, that of the FLT3-ITD, which we know FLT3 when activated, activates AKT, and so it was no surprise that in the setting of FLT3-ITD, there was not a high incidence of activated FOXO, of high FOXO. Now, this idea of FOXO expression being somewhat either high or low in AML was also associated with different gene expression, suggesting that it was not just the expression of FOXO, but its activity that was broken down into these two major categories of AML. And the FOXOs, by the way, there are multiple different FOXOs. We're going to switch between talking specifically about FOXO3, the predominant FOXO in hematopoietic stem cells, and the broad group of FOXOs which were deleted in our animal model. So broadly, we can say that that AML is in two categories: those with high FOXO activity, about 40%, and low FOXO activity, or phosphorylated FOXO, at about 60%. So we focused on those we wanted to focus on those with the high FOXO activity, and to do this, we wanted to address it in an animal model that was tractable to both functional and genotypic analysis. And so AML has the advantage, with its known different genotypic abnormalities, of giving us an opportunity to take a mouse model with mouse cells that you could isolate and then transduce with human leukemogenic alleles. And in this context, you can transplant the cells into the mouse, and you end up getting what appears to be a disease that somewhat resembles that of the human, where you get an increase in the number of leukocytes in the peripheral blood, as seen on the left, with abnormal appearing cells in the peripheral blood, and massive splenomegaly with infiltration with these leukemic cells. And this is a highly penetrant phenotype. It's a very useful model for AML. It's really developed by Scott Armstrong's lab and has been used now by us and many other laboratories. So we asked then, in this genotype, in this model, where we're using, again, a human leukemogenic allele, what happens to FOXOs? So in order to assess this, we wanted to look at what we thought would be the parts of the pathway, that is activated AKT present or not. And what we found in the setting, of either the normal myeloid progenitors or leukemic myeloid progenitors, that there was a difference. That in the setting of the myeloid progenitors, the leukemic progenitors, you actually had decreased in the amount of serine 473 phosphorylated AKT. And similarly, if you look at, one of the products of the activated AKT, that of S6, looking for its evidence of it being activated by virtue of phosphorylation. Similarly, in the setting of MLL-AF9 leukemia, you ended up with a blunted phosphorylation of S6. So it does seem that in this mouse model of AML, you also see a low level of AKT activation. And asking whether or not this is also borne out with FOXO. As I mentioned before, FOXO, when phosphorylated, is inactive because it's unable to enter into the nucleus. It's in the unphosphorylated state that it enters the nucleus and acts as a transcriptional regulator, both positive and negatively. So we then looked to see whether or not FOXOs were in the nucleus or in the cytoplasm in the context of this mouse model of human leukemia, and found that indeed, in the context of leukemia, there was a great deal of nuclear localization, whereas under normal conditions, it's usually in the cytoplasm. Looking then at whether or not there was evidence of activation of particular pathways, that is, expression of genes that were either activated by FOXOs or repressed in the context of the population of cells that were leukemic in our animal model, there was correspondence indicating that FOXO was indeed increased in its activity in our leukemia model. So then asking whether or not, if we now delete FOXO, does this have any impact? We could put this into the background of an animal that had conditional deletion of a number of different FOXOs, and then look to see what the deletion of FOXO would do to the leukemic phenotype. And we found that if we do that, you see a reversion of the splenomegaly that was present in the animals, and you end up with very different leukocytosis, where the leukocytes that are seen in the control animals are much mitigated in the setting of the deletion. Similarly, if you look at overall survival, you see an improvement with about 20% of the animals having a long-term survival. Still a big drop in survival, which I'll talk about in a few minutes. But basically, you could see that there were some animals that had evidence of essentially elimination of their leukemia. So we asked then whether or not this, again, animal model has correspondence to humans. So now we wanted to look at human AML in greater detail, not just the original gene expression profiles, and to look at whether or not this was restricted to the MLL genotype that we'd used for our model. So what we did is initially look in cell lines to see whether or not we could, first of all, knock down FOXO3A, which we could. And then the question is, what happens if you put this into human AML cell lines? We found that it reduced the growth of those that had MLL-AF9. These are the different ways of knocking down FOXO. But interestingly, it was not restricted to the leukemias that had the MLL-AF9 genotype, that in those that were cytogenetically normal, as well as some with the APL 1517 translocation, also blunted cell growth. And then we got human AML samples from patients where you can occasionally grow out cells. We did on 2 occasions, and we showed that with the knockdown of FOXO3, you end up having a decrease in the ability of these cells to culture in methylcellulose. So, just briefly, FOXOs are active in a reasonable proportion of AMLs. That AKT is diminished in the animal model of AML, and that this is something that is associated with high FOXO, and if you deplete FOXO, you end up depleting the AML. This is also seen in human AMLs. So now the question of whether or not it impairs leukemia-initiating cells, getting back to this idea that what we have learned about cell populations and normal stem cells can be applied to leukemia. Well, does it deplete the leukemia-initiating cell that has been a putative leukemia stem cell demonstrated by John Dick? And what we did was basically take this subpopulation of cells that could be fractionated based on features to suggest that this is a correspondent primitive population. We showed that those numbers were diminished in the setting of the inactivation of FOXO. And if we look at how these cells perform, if you do a limiting dilution now, taking smaller numbers of the leukemia cells and transplanting them, if we just transplanted the leukemia cells, you can see that it's lethal at different orders of magnitude of the transplanted cells. But if you now deplete FOXO, you shift that up by almost 2 orders of magnitude. And so you end up reducing the leukemia-initiating cell frequency by about 15-fold by the depletion of FOXO. So it does suggest that the leukemia-initiating cell, the leukemia stem cell frequency is diminished in this context. And why is it that FOXO has this effect on leukemia? When we look to see what happened when we knocked down FOXO in cell lines, we found that those cell lines now went from poorly expressing markers of differentiation to increasing their expression of markers of differentiation, that they acquired a morphology, it's hard for you to see, but basically went from a blast-like morphology to having more of a mature morphology by histology. And if we look at their ability to now undertake functions like phagocytosis, they went again from that of a poorly functioning cell population to a more functional population, and that this was associated also with the acquisition of terminal differentiation phenotype that included spontaneous cell death. So these were then validated in 7 other human leukemic lines. So it would suggest that the FOXO is preventing the cell population from being able to differentiate, and then when you deplete FOXO, the cells will undergo a maturation process that leads to their terminal differentiation and death. Now, we wanted to again validate that this was true in primary AML, so taking those patient samples, we showed that if you now knock down FOXO, they decrease their ability to form colonies, but they also increase their expression of markers of maturation, and they had a morphologic appearance where they acquired things like cytoplasmic granules and some maturation in morphology. So in general, we've shown then that FOXOs are required for the leukemia-initiating cell function, and that it's required to maintain a differentiation blockade in AML. But we had this issue of cell populations that in vivo, some of the animals were able to survive longer term, but there was a fair amount of animal death that still occurred despite the deletion of FOXO. And we wanted to understand this to essentially address the question of what provides FOXO resistance. And so when looking at this population of animals that had died despite deletion of FOXO, we found a very interesting phenomenon. That is, that when we look back at what might be different in the in humans that do or do not have activation of FOXO, what we found is that there's a differential regulation that's inversely, an inverse relationship between FOXO and c-JUN gene expression. And so we looked to see whether c-JUN was altered in these animals. And indeed, we found that there was, again, an inverse correlation between expression of FOXOs and c-JUN, where low FOXO, high c-JUN, higher FOXO, lower c-JUN. And so when we looked in these animals, taking now the cells from animals that were dying of the MLL-AF9, we found that indeed in those animals, c-JUN was increased in its phosphorylation by Western blot, suggesting that indeed there was activation of the c-JUN pathway. So c-JUN in its phosphorylated form is activated. It gets phosphorylated by the JNK or JUNK kinases. Their phosphorylation results in these events. So we looked in the leukemias to see whether or not we had evidence of low FOXO corresponding to increased phosphorylation of c-JUN. We did. And we also saw phosphorylation of JNK, suggesting that this pathway was indeed the one that potentially could lead to this altered sensitivity. So we know that there are JNK inhibitors available. So we applied a JUNK inhibitor to the cells that had been resistant to FOXO depletion to see whether or not that could lead to cell depletion of AML. So we added the JNK inhibitors, and in conjunction with knocking down of FOXO, we end up getting a loss of cells in vitro. The cells are no longer capable of undergoing proliferation. In addition, the cells undergo a very rapid and more full differentiation that is again associated with advanced apoptosis. So the cells undergo a more rapid and complete differentiation program and apoptosis in the context of the combination of FOXO inhibition and JNK inhibition. So we think then that we've gotten to a point, I'm ahead of myself, where we can look at this complex genotype of different AMLs and now try to impose something of a simpler way of viewing them through the lens of what we know to be important in the regulation of normal self-renewing cells, and that is through FOXO signaling. And in this case, we found that there's a subpopulation that has high FOXO. These are generally associated with low JNK activation, but in the absence of, and in the other population, there's low FOXO with high JNK, and we found that in the depletion of FOXO, you end up with high JNK being able to enable these cells to be FOXO resistant. So we think that this has given us an opportunity, I can't go back, to think about AML in more broader terms. We think that may be more tractable to drug development, in that if you think about now targeting a signaling pathway or signaling pathways, we may have an opportunity to be able to intervene in ways that can affect much broader categories of AML rather than trying to individually focus on particular genotypic alterations. And so with that, I want to close by thanking the people in my lab. You can see a very dour group, but in particular Steve Sykes, who I think is standing on higher steps, but the guy does go about 6'4", and so he always stands out in a crowd, but he's done a particularly good job with this particular project. And again, this has been a real demonstration of his capability to really apply this idea of understanding things in normal stem cells as a way to try to interpret issues in cancer and potentially come up with novel ways to approach them. Now, I will say that as a cell biologist, much of this, the genetic manipulation often seems a little beyond me, so a colleague has provided me with a primer on genetics that I thought I would share with you as a way to close. And I'll thank you for your attention.
Thank you, Dave.
Thank you.
Maybe I could start off with a
Sure.
questions. Is this on? Is it working? There's a link between redox balance and differentiation of leukemia cells
Right.
and driving loss of self-renewal with differentiation. How does FOXO relate to that science?
Yeah, very much. So FOXO is thought to be an important component of the response to an oxidative stress. It does regulate the expression of many of the genes that are important in being able to respond to and control elevated levels of reactive oxygen species. And so we think that that's probably the way it's working, but we haven't specifically gone to those targets.
Yeah, thanks. Any questions from the audience?
Hey, John.
David. I was curious, if you take the kind of findings to the nth degree, does it suggest that in the cancers that have, or the leukemias that have high FOXO activity, that is maintaining it in this stemish type of state, that actually activating PI3 kinase induces differentiation of those leukemic cells?
Well, so we are in the process of testing that question, but it is a reasonable hypothesis that if you now increase the activation of AKT, that you would downregulate FOXO as a way to induce differentiation. Right? And so it is something that we are trying to test at this point. So we've done genetic modifications to really eliminate FOXO. I'm not sure you can ever get to that level of depletion with an activation of AKT, but it is one of the questions that this raises, absolutely.
Other questions? Bruce?
David, does this biology just apply to the subset of cells that are stem cells in the leukemic population, or is the effect of FOXO seen also in the more mature and larger population of circulating leukemic cells?
Well, it's a great question, and we think that it actually applies to the broader population because, for example, when we take the primary patient samples, while we do have some, if you will, selection of subsets of cells by those that will grow in vitro. They are clearly affected by alteration of FOXO. Those may represent some enrichment of a stem-like population, but I doubt that it's really down to just that core element of cells. So it's hard to know because in the mouse model, it looks as if when we look at the leukemia-initiating cell, those do seem to be most sensitive, but I think it's not restricted just to that population.
Other questions? David, just one quick question. In the low FOXO leukemias, does it make sense there to use the JUNK inhibitors?
Well, we would suggest that that's true, but I think JUNK inhibitors by themselves have not had a very terrific experience. And so it may be that it's really in the context of FOXO, which will be extremely difficult to inhibit. I mean, it's a hard to drug target, but the combination of the two, we would think, would be particularly productive.